So as Professor Carter introduced, today I would like to have a talk about self-assembled nano device made from block copolymers, which we are using this for as a nano carrier for gene and drug delivery. So first I would like to briefly mention about the concept of targeting therapy by utilizing nanocarriers. Concept is quite simple. So when you inject a drug through intravenous route, usually drug spread all over our body and eventually cause some severe side effects and decrease the bioavailability of drugs. So the most simple answer to solve this problem is to develop nanocarriers which selectively accumulate to the target site and put this drug or bioactive substances inside of the nanocarrier systems. Then eventually you have a high accumulation on, to the target site with decreasing side effects and increase the bioavailability of drugs. So the concept is quite simple. However, make this into reality, you have to overcome many tough issues. So here I summarize several issues which we should overcome for the successful drug and gene delivery. The first thing we should think about is longevity in blood circulation. So when we inject the nanocarriers or particles into our blood compartment, usually these particles can be recognized as foreign body and easily captured by a group of cells called reticuloendothelial systems, abbreviated as RES. These are a group of cells settled in organs such as liver and spleen. So to avoid this recognition by RES, we have to engineer the surface of the nanocarrier system to be totally biocompatible or stealth properties. So that is the first thing we should think about. The next issue is extravasation, because when we would like to target solid tumors, the tumor cells locate outside of the vasculature. So definitely the nano device, nanocarrier needs to translocate from inside of the blood circulation into solid tumors and penetrate deeply into the mass of the tumors. So in this regard, the size, as I mentioned later, the size of the nanocarrier is quite important. Should be less than 100 nanometers, or if you want to make this to have a high penetration ability into hypovascular fibrous solid tumors, you may be better to tune the size of the nanocarrier may be less than or almost 30 nanometers. And when your payload is a cytotoxic reagents, in this way you can accumulate cytotoxic reagents in solid tumors and maybe drug released from this nano device and directly can penetrate into tumor cells. However, if you want to deliver the gene or oligonucleotides, such as siRNA, unfortunately, these oligonucleotides and gene cannot directly penetrate cellular membranes. So in this case, you definitely need nanocarrier itself to find the target cells and, like a Trojan horse, internalize into target cells, and also by controlling the intracellular trafficking and to deliver the, for example, in case of the gene therapy, deliver gene directly into final target nucleus. So eventually you can have achieved the targeted therapy. So by overcoming all of these issues, then you can deliver bioactive substances in a desired site of the body with desired timing. So of course, there are many approaches to construct this kind of nanocarrier systems. And the strategy which we have been working on is self-assembly of synthetic polymers, most typically synthetic block copolymers. I think most of you are the chemistry background, you are familiar with the terminology of block copolymers. If you are not familiar with polymer chemistry, it's a kind of the polymer which conjugate to different blocks or segments with a very different chemical nature. So this part maybe is hydrophobic blocks, and this green segment should be hydrophilic segment, and put this into aqueous entity, then this block copolymer spontaneously self-assemble into core shell structures, which we called polymeric micelles. And maybe you are familiar with micelles made from low molecular weight surfactant molecules or soap micelles. But soap micelles usually not stable. So if you put this soap micelles in a very diluted condition, it easily dissociates. However, in case of the polymer, this is a high molecular weight compound. So if you design the structure properly, then you can expect a very high association force or a very strong cohesive force. So eventually, these polymeric micelles can have a very high stability even in a very diluted condition, or in other words, very low critical micellar concentrations. So definitely this is a very good advantage of utilizing this system as a drug carrier. That means that even after injection into the bloodstream, of course that is a very diluted condition, still you can expect that micelle can keep their original shape and transport drug into target tissues. And the core of the polymeric micelles are the nano container of payloads, drug or gene, which is totally surrounded by dense layer of tethered hydrophilic polymer segments, most typically polyethylene glycol. So because of the hydrophilicity and very good flexibility of polyethylene glycol chain, now you can keep the interaction with blood component quite minimum, or minimum interaction with blood components, for example, proteins and cellular surface molecules. And eventually, this system becomes totally stealth or biocompatible in bloodstream. And the size range of the polymeric micelle, of course, you can tune in the range of sub-100 nanometers, most typically 50 nanometers. And you can make this system in a totally regulated thermodynamical status to make this micelle with a very narrowly distributed characteristics, as shown here. So this is a typical atomic force microscopy of polymeric micelles, and this is a 100 nanometer scale. So you can see each of the micelle has around 50 nanometers with very narrow distribution. And let me remind you that this size range of 50 nanometer is almost the same size range as Mother Nature's nanocarrier systems, virus and lipoproteins. So that means that you can expect a very high extravasating abilities. And of course, if you want to make this to have active targeting properties, you can conjugate some ligand molecule on the periphery of polyethylene glycol chains. And chemical structure of core is quite important, because we would like to accommodate the payloads with a very different chemical natures. And also, the core chemical structure should be biocompatible, or maybe better to be biodegradable. So from that standpoint, polyamino acid is quite promising, because amino acids, it's a component of proteins. So you can expect the polyamino acid to be biodegradable, biocompatible, and by changing the chemical structure of side chain, you can make this from hydrophobic to hydrophilic, anionic to cationic. So that's the reason we choose polyamino acid as the core forming segments. So as shown here, already our developed polymeric micelles from polyethylene glycol polyamino acid block copolymer for different formulations. I mean that the different cytoplasmic regions are already in phase 1 and phase 2 clinical trial, not only in Japan, but also in Asia, USA, UK, and France. Paclitaxel, cisplatin, SN38, that is active metabolite of irinotecan, dahaplatin, and epirubicin, that is in preclinical development, and this one here, this is an isomer of very well-known anti-cancer drug, doxorubicin or adriamycin, and we make this system to have a pH-sensitive polymeric micelles. So I will show you the one example of paclitaxel because this is it's in the most progressive stage of micellar carrier development. That is just complete the phase 2 clinical trial and now in the preparation to phase 3 clinical trial. So this is a chemical structure of paclitaxel, very complicated organic compound which is not immiscible with water. But paclitaxel can be soluble in organic solvent like dichloromethane. So we modify the side chain of polyamino acid or block copolymer to make system to be a little bit more hydrophobic, and eventually this PEG hydrophobically modified polyamino acid block copolymer can also dissolve in dichloromethane with paclitaxel. Then put this mixed solvent into water and emulsify and evaporate the dichloromethane. Eventually, we can stably accommodate paclitaxel molecule in the core of the polymeric micelles. And this polymeric micelle drug, paclitaxel, shows a very high anti-tumor efficacy, as shown here. This is a result of the animal experiment, and this is a relative tumor size. And PTX, that is a conventional formulation of paclitaxel called Taxol. So compared to Taxol, the NK105, that is a development code of polymeric micellar drugs, compared with the same dose, always the micellar system shows a much higher anti-tumor efficacy. And by increasing the dose to 100 milligram kg, we clearly observe the total regression of the tumors. And this is a result of animal experiments. And here is one of the examples of the clinical trial. That is a treatment of the metastatic pancreatic cancer into liver. So these black spots corresponding to the metastasis of pancreatic cancer in the liver pre-treatment. And after two and a half months of treatment by polymeric micellar drugs, all of these metastasis region are totally disappeared. So it shows the very high efficacy of this paclitaxel micellar drugs. Then I will show you more detail about the dahaplatin polymeric micelles, because recently we found out by this formulation we can treat the drug-resistant tumors. The treatment of drug-resistant tumor is a very important and very difficult in cancer chemotherapy. But by utilizing this polymeric micellar dahaplatin, we can overcome the problem of drug resistance. So here is a chemical structure of dahaplatin. That is a platinum complex. And because of this cyclohexyl ring, the dahaplatin has a very low solubility in water. That is not soluble. So to solve this problem in clinical situation, this prodrug of dahaplatin called oxaliplatin are used in clinics. So because of this oxalic acid structures, now you can increase the solubility, water solubility of the drugs. However, still oxaliplatin has many problems. Now this oxalic acid moiety has a neurotoxicity, and anti-cancer activity is not always interesting. That means that anti-tumor efficacy is not enough to treat intractable tumors. So we want to increase the anti-tumor activity of dahaplatin and decrease the side effects. We load dahaplatin into polymeric micelles. The procedure is quite simple. So first, we mix this dahaplatin and polyethylene glycol polyglutamic acid block copolymer in aqueous solution. Then it spontaneously undergoes the ligand exchange reaction and form this carboxylate complex of dahaplatin at the core of the polymeric micelles. So the reaction of dahaplatin with carboxylate groups of PEG polyglutamate block copolymer spontaneously trigger the self-assembly of block copolymer into polymeric micelles. And size is around 30 to 40 nanometers with a very narrow distribution. So this structure is quite stable in aqueous solution. But once you put this again into physiological condition with an increased sodium chloride concentration, then opposite reaction takes place to release the active compound. However, always it shows an induction period of about 10 to 20 hours. So during this induction period, this polymeric micelle accumulate to the solid tumors, and then after that, the slow release of dahaplatin occurs. So by formulating dahaplatin into polymeric micelles, we can significantly improve the biodistribution. So here is a platinum concentration in the plasma, and this is oxaliplatin. Oxaliplatin quickly excrete from blood circulation. On the other hand, dahaplatin micelle, even after 48 hours, still 10% of initial dose stably circulate in the blood compartment. And because of this long circulation, we do observe the very high accumulation to solid tumors, as shown here. So at 48 hours, the difference is more than 20-fold. So when you compare this by area under curve, that is a cumulative accumulation, the difference is more than 100 times. So how come this increased accumulation occurs? This is explained by the effect called enhanced permeability and retention effect, EPR effect. I will explain briefly what is the EPR effect. So this is a cartoon of the blood capillary, and the upper part of this capillary shows a capillary vessel wall structure of capillary in normal tissues. In normal capillaries, always covered by a very tight endocellular layer, which is backed by the basement membrane. So because of this tight structure, the polymeric micelles with a 50 nanometer diameter cannot penetrate into normal tissue. And this is good because in this way you can decrease the non-specific accumulation of drug to normal tissues. However, the small drugs still can diffuse through this barrier into normal tissues and cause the problem due to non-specific distribution. Now, the lower part of this capillary shows the structure at tumors. The tumor capillaries has many openings between endocellular layer. That means the endocellular layer is not continuous. Always there is openings. And basement membrane structure is also not continuous. And because of this leaky structure, the long circulating carrier has a good opportunity to accumulate into solid tumors. So because of this kind of the transport phenomena, we can have a very high accumulation as high as 20-fold increase. So I will show you this process in movie. I'm sorry, this is in Japanese. This is vessel. And this is a normal vessel, but still normal vessel, there are small pores which small drugs can penetrate. However, polymeric micelles are large enough, so they cannot penetrate into normal tissues. And in this way, you can decrease the side effects. However, this is the structure of the capillary at the tumors with a large openings, and micelles can penetrate through these openings into tumor tissues and penetrate into tumor tissues and internalize into tumor cells by a process called endocytosis. Because micelle is too big, so it cannot diffuse through the plasma membrane, but alternatively, the micelle can internalize into the tumor cells, totally covered by the part of the plasma membrane, so which called endosome, and transport into perinuclear region. So because of this process, we can overcome the drug resistance. So here is the results. This is the treatment of auxiliary protein resistant tumors. So now auxiliary protein has almost no efficacy. However, still micelle shows a very significant suppression of tumor growth. And because of this high anti-tumor efficacy, this is now in phase 1 clinical trial. Of course, the original tumor and drug-resistant tumor both has a same in terms of the biodistribution and pharmacokinetics of polymeric micelles. So this is not due to the difference in EPR effect. This is due to the intracellular trafficking route of single cellular labels. Indeed, when we observe the IC50 value, that is a concentration of drugs to kill 50% of cultured cells. The IC50 value, in case of auxiliary protein, there is a 10 times increase for resistant tumor cells. That means that the auxiliary protein reduces the efficacies because of the drug resistance. However, in case of the micelles, this value is no change. Means still micelle shows a high activity against auxiliary protein resistant tumor cells. So how come explain this mechanism? So I will show you this in the cartoon again. So this is the drug-resistant mechanism of platinum drug by drug-resistant cancer cells. So anti-cancer drug dahaplatin is small molecule, so directly internalized into cytoplasm. But there is an overexpression of defense proteins in this kind of the drug-resistant cells. So the auxiliary protein cannot penetrate into final target nucleus. On the other hand, the polymeric micelles, the nanoscaled Trojan horse, internalized into cancer cells by, as I mentioned, process of endocytosis and accumulate in perinuclear region, which trigger the micelle dissociation and directly accumulate to the nucleus by bypassing defense proteins. So this mechanism is quite, I will explain more detail about this mechanism. So in case of the, as I mentioned, the auxiliary protein can diffuse directly into cytoplasm by simple diffusion or through CTR, that is a copper transporter, and inactivated by overexpressing proteins like methionine synthase and metallocyanine. Now, in case of the polymeric micelles, internalize into endocytosis and transport into perinuclear region. So why selectively the drug release occurs in perinuclear region? This is due to the pH and salt concentration difference. So in the early endosome, that means that endosome near cellular membrane has a pH of this pH and this sodium chloride concentration. But when endosome transport into perinuclear region, there is a decrease of the pH and increase of the sodium chloride concentration. And when we carried out a drug release study corresponding to these two conditions, the result is very striking. In the early endosome condition, there's almost no release occurs, no drug release occurs. However, in the late endosome condition, we do observe a very fast, quick release of DAHA protein from polymeric micelles. So that means that in the early endosome, drug cannot release from polymeric micelles, but after reaching to the perinuclear region, there is a start of the release of the drug and quickly accumulate into final target nucleus. So to confirm this hypothesis, we prepared this labeled block copolymers. Here we labeled the distal end of PG of block copolymer with green fluorescence dye and conjugate red fluorescence dye in the distal end of polyglutamate segment and make micelles. So in this case, the red fluorescence is totally quenched in the core of the polymeric micelles. So if the micelles are in intact condition, we observe only green fluorescence. However, when micelles starts this integration to release the drugs, then we should observe red fluorescence. So we should observe the merged color of red and green means maybe we should observe yellow or orange colors. So by utilizing this kind of the probe molecule, we confirm the selective release of auxiliary protein or DAHA protein in perinuclear region. So I will show you this result in the movie. So this is a tumor cells, and you see green fluorescence means internalization of intact micelles, and this is the time. And when time reached about 50 hours, you see orange colors inside of the cells, corresponding to the time, or corresponding to the release of DAHA protein, synchronized with the disintegration of polymeric micelles. But still, this is an in vitro study, so we move into in vivo. But we'd like to see what's going on inside of the animals. So to see this, we utilized a rapid scanning confocal microscopy just developed by Nikon. So this instrument it has a rapid scanning mode, the video rate. So it means you can have a very clear image of confocal pictures, even animals are breathing. So before one breathing, it scans all the regions. So you can get a very clear image of living tumor cells. So we prepare the tumor in the subcutaneous of the animals and observe this by laser scanning microscopy. And this is one example. This is the whole tumor with green fluorescence, expressing the green fluorescence proteins. So each green dots corresponding to the single tumor cells. And this is a three-dimensional stack image of tumors inoculated in the subcutaneous of the experimental animals. And these yellow vessels are the capillaries. So by utilizing this picture, we now can follow what's going on inside of the tumor tissues. Now, here again, I will show you that the green fluorescence is from intact micelles. And when micelles are in a decayed process, you should observe red fluorescence. So just after injection, you see green fluorescence in the capillary, and these black spots corresponding to the erythrocytes. That means that the intact micelles stably circulate blood capillaries in tumor tissues. Then after 12 hours, still you see the green fluorescence in the blood vessels, means still the intact micelles stably circulate in blood capillary. But if you see in a perpendicular direction, you can see each of the cancer cells with the inside of, you see yellow or orange fluorescence. which corresponding to the disintegration of the polymeric micelles. So this is the first direct observation that of nanotrojan pores really can penetrate into tumor tissues and internalize into tumor cells and release drugs in the perinuclear regions. So in this way, we can overcome the drug resistance. And this is an example of utilizing dahaplazine, platinum cancer drugs. But of course, this strategy also applicable for the delivery of oligonucleotides or different active chemical compounds. Now, we would like to also we would like to treat not only the drug-resistant tumor cells, but next challenge is to treat the intractable cancer. Most typical example is pancreatic cancer. Why pancreatic cancer is intractable? One of the reasons is that the hypovascular properties with very fibrous tissues. So here is a CT image of the patient with pancreatic cancer. Even utilizing the imaging reagents, it's very difficult to diagnose pancreatic cancers because of the limited penetration of imaging reagents. And eventually, the diagnosis is very difficult, and chemical and chemotherapy of pancreatic tumor is quite difficult. So to challenge the treatment of pancreatic tumor, we are focusing on the effect of the size of the polymeric micelles. So we prepare the polymeric micelle with a different size from 30 to 80 nanometers. And also we confirm these three micelles shows a totally same blood circulation behavior. All of these three micelles shows a very long circulation properties. Then we utilize this for the treatment of the tumor. So first, we treat the colon cancer model, which is characterized by very hypervascular tumors. So in this case, there is no difference. Both 30 nanometer, 80 nanometer micelles quite significantly suppress the tumor growth. Then we see for the pancreatic tumors. So before I show the results, I will explain a little bit about the characteristics of pancreatic cancer, characterized by hypovascularity and thick fibrosis. So here, this blue region, this is the nest of the tumor cells. And you see, maybe you see some red spots here, and this corresponding to vasculature. And this dark region is fibrous tissues. So very important point is that the vasculature is not exist inside of the tumor nest. Vasculature only locates in the fibrous region. So even if your carrier systems extravasate from vasculature, still the carrier needs to travel through this fibrous tissue to reach the final goal, tumor nests. So this is a very difficult for the particles. Then this is the results. So result is very significant. The 30 nanometer micelles still works quite nicely. However, 60, 80 nanometer micelles have no efficacies. So this is a very critical phenomena depending on the size. So to confirm, so we would like to confirm this due to the difference in penetration. We again utilized in vivo laser confocal microscopy. In this case, we label the 30 nanometer micelle with green fluorescence and 80 nanometer micelle with red fluorescence, and mix these two micelles and inject. Now, the merged color of green and red is yellow. So eventually, the inside of the capillary is totally color yellow. But if you see the tumor tissues, it's green color. Means only 30 nanometer micelles can penetrate. So where is the red micelles? Red micelles can extravasate. However, it's stuck on the periphery of the capillaries. That means that both 30 nanometer, 80 nanometer micelle can extravasate. However, 80 nanometer micelle cannot penetrate into this fibrous pancreatic tumors. And penetration can be observed when you decrease the size of the micelle to 30 nanometers. And we further confirm this by microsynchrotron radiation X-ray fluorescence in spring 8 by utilizing the high energy electron beam. So here is the tumor observed by this microsynchrotron radiation X-ray fluorescence. So we can identify the tumor location from the signal of ion. Because as I mentioned, in the tumor nest, there is no blood. No blood means no erythrocyte and low ion concentrations. So this is a tumor nest. And this is a distribution of platinum. In 80 nanometer micelles, there's almost no platinum in the tumor nest. However, in case of the 30 nanometer micelles, we do observe the very high signal even in the tumor nest. So from this explanation, this experiments, we confirm that if you precisely tune the size of the micelles, you can expect that can penetrate into tumor nest. So the nanotechnology is very important for this kind of the drug delivery systems. So further, by incorporating the imaging regions in this nanocarrier systems, we can make this Theranostic Nanodevice. That means a single platform device of therapy and diagnosis. And eventually, we can see the accumulation of carrier systems into tumors and also monitor the device accumulation and the response to the therapy. So we prepared this kind of Theranostic Nanodevice based on our dahaplatine micelles. The chemistry is quite simple again. When we mix this polyethylene glycol polyglutamic acid block copolymer with dahaplatine in the presence of gadolinium DTPA. Because of this the gadolinium DTPA has 5 carboxyl groups, and one of the carboxyl groups is almost in the free condition. So we can expect that through the platinum bridge, we can incorporate gadolinium DTPA in the core of the polymeric micelles. And not only the incorporation of gadolinium DTPA into polymeric micelles, we do observe the enhanced T1 reactivity. That means that increased sensitivity. The reactivity increased more than 20 times, means by incorporation of gadolinium DTPA, we can even increase the sensitivity of gadolinium DTPA. So because of this high loading efficacy, we do observe the accumulation of dahaplatine and gadolinium DTPA in colon cancer, as shown here, compared to free gadolinium DTPA. And eventually, we had a very nice imaging results. So here is this is a tumor region. By free gadolinium DTPA, there is almost no increase in the signals, even after 240 minutes. But the tumor treated with this Theranostic micelle, just after 5 minutes, you already observe some the intensified spots, white spots, and after 240 minutes, the whole tumor becomes totally bright. That means by utilizing this kind of system, you can have a very nice imaging of tumors. And this is a subcutaneously xenograft model. So we would like to see this system also works for the diagnosis of orthotopic model of pancreatic tumors. So we prepare the orthotopic tumor in the pancreas and inject the micelles and make a specimen of tumors. This is a tumor region, and this is a normal pancreas tissue, and brought this again into spring 8 to see the distribution of platinum and gadolinium in tumors. Now, in this case, we identify the tumor region by increased signal of potassium, because potassium is a cofactor of pyruvate kinase, the enzyme overexpressed in pancreatic tumors. So we can see this region is tumors, and these are the tumor nest with a very high potassium concentration. And this is a distribution of platinum, and this is a distribution of gadolinium, and both of the distribution is nicely corresponding to the distribution of potassium, and this is a direct evidence that by utilizing this system, we can accumulate platinum and gadolinium together into orthotopic pancreatic tumor models. And eventually, we do observe the very nice imaging and tumor treatment efficacies. So this region here, this is a pancreatic tumor, an orthotopic pancreatic tumor, before the injection. And after injection, you see some increased intensities. And after 18 days, this pancreatic tumor shrinked because of the efficacy of dapagliflozin. And we confirmed this after sacrificing the animals from the relative tumor volume and weight of pancreas. So in this way, we can now have a Theranostic Nanodevice which simultaneously diagnosis and therapy in single platform device. Now, maybe I have still maybe 10 minutes left. Then I would like to explain about our gene delivery project. So usually in the gene therapy, people use a virus. This is adenovirus. This is a Mother Nature's nanodevice. In here, the core, the DNA is nicely packaged, which is surrounded by capsid proteins, and size is around 80 nanometers. Of course, virus is a very high transfection efficacy, but still several problems, mainly from safety issues. So we'd like to prepare kind of artificial virus by self-assembly of plasmid DNA and block copolymers. So plasmid DNA had an anionic nature. So we just mix this with the oppositely charged block copolymer of polyethylene glycol and polycation to make an ion pair. And in this way, plasmid DNA is totally covered by PEG and induce the phase transition or condensation. So the supercoiled extended DNA conformation shrinked into rod or toroid structures. So that is very interesting to see this kind of a very regulated folding. And because the plasmid DNA, that is a double-stranded DNA and coiled structures. So that is a very, very rigid molecule. So usually to fold this rigid molecule into packaged into rod is not easy. So we explore the mechanism and found out that this process is kind of the very regular folding in quantized folding model. So when plasmid DNA interact with block copolymer, first it transforms into rod structures, and this rod folded into the center to this structure, the one folding, single folding, and double folding, and triple folding. So it's very quantized, regulated folding process. How can we confirm this folding process? This is from direct measurement of long axis of each DNA rods and see the distribution. And distribution is very interesting. It's totally quantized, 608, 304, 203 nanometers. And this 608 nanometer exactly corresponding to this initial rod of plasmid DNA. And 304 is half of the 608. So this is a one folding structure. And 203 is two folding structure. And 152 is three folding structures. So it's quite regulated folding of plasmid DNA. And even by utilizing the dynamic AFM, we can see the bending process of plasmid DNA, as shown here. And if you are lucky, you can see the DNA almost folded into 2 folding structures and 3 folding structures. And this single plasmid DNA is totally covered by polyethylene glycol. So then next, we would like to see the thickness of polyethylene glycol layer cover the plasmid DNA. So to measure the PEG thickness, we concentrate this DNA loads and take the cryo-phase contrast TM, as shown here. So this is a cryo-phase contrast TM. And in here, we utilize a block copolymer. 1239 means that the molecular weight of polyethylene glycol is 12,000. And polymerization degree of lysine unit is 39. And this is a spacing, and average is 80.6 nanometers. This is very interesting because we utilize PEG with 12,000. And usually, the PEG 12,000 random coil diameter can be calculated as 9.36 nanometers. When you divide this 18.6, it is 9.3. So means that the plasmid DNA is covered by random coiled PEG, mushroom conformation. Then we decrease the polylysine unit from 39 to 19. So in this way, because of the decrease of the polymerization degree, means you need more polymer chains to neutralize DNA molecules, and eventually increased number of polymer chain means there should be the increased density of PEG. And when PEG density increases, there should be the stretching of PEG molecules. And eventually, the average spacing increased from 18.6 to 24.9 nanometers, which shows that in this case, PEG shows a slightly stretched conformations. So already there is a previous paper about the relationship of the PEG height and density of PEG molecules. And in this region, the PEG shows a random coiled mushroom conformation. And when increase of the density, it transfer into crossover region, and finally adapting to scaling law, brush-like structures. And this density of 1239 and 1219 corresponding to this region. This means that in this rod-like Micelle structures, the plasmid DNA is covered by mushroom or slightly stretched PEG layers. So because of this covered by PEG, we can expect a very increased biocompatibilities. So I will show you demonstrate this again by in vivo laser confocal microscopy. So we see the circulating polyplex in blood capillaries. Now in this case, we fluorescence labeled the platelets in the green fluorescence. So the platelet size is 1 to 4 microns. On the other hand, polyplex size is 100 nanometers, much smaller than platelets. However, in case of the DNA polyplex without PEG, what we observe is that the red aggregates almost the same size as platelets. That means that without PEG, after injection, the polyplex instantaneously form large aggregates, and you can see some yellow, the regions means stick to the platelets and may induce clot formation. So that means that you cannot inject this polyplex because this may cause big problems. However, when we cover the polyplex with dense PEG layer, the result is totally different. You see here the platelets and the background is red color, no aggregates. So in this way, by cover the polyplex with PEG, you can safely circulate the polyplex in a blood circulations. So in case of the gene delivery, not only the systemic circulation, another issues, that is endosome escape. Because gene vector, whatever non-viral or viral vectors, both internalize the cell by endocytosis and captured in endosome, and finally digested. So definitely, we need some mechanism to endosome to escape from endosome. So Mother Nature's virus is very clever. The virus has a very special proteins, peptides on the surface, and at pH 7, the membrane disruptive region is hidden inside. And in the acidic compartment of endosome, this membrane disrupting region exposed outside and disrupt the integrity of endosomal membrane to facilitate the endosomal escape of viral particles. So if we can integrate this kind of function into our non-viral vector systems, we can expect a high endosomal escape properties. However, the issue is that this membrane disruption should selectively occur only inside of the endosome. If this membrane disruption occurs in the plasma membrane, it causes severe damage to living cells. So we take quite a long time to identify the good structures, and we prepared many different types of polycations from polyaspartate. And finally, we found out these structures, which shows that this is a polyaspartamide with ethandiamine unit as a side chain. Because the one reason is that by utilizing this system, we can confirm the very high efficacy of endosomal escape, as shown here. So this is a single cell. When we complex the plasmid DNA with conventional polycation, polylysine, without any endosomal escaping properties, and here, in this case, the dextran, green fluorescence from dextran endosomal marker, and red color from plasmid DNA. And both dextran and plasmid DNA still captured inside of the endosome in case of the polylysine polyplex. However, the plasmid DNA complex with PASP-DET, which is the ethandiamine unit, after 24 hours, you clearly observe the diffusion of both plasmid DNA and dextran into cytoplasm. And another important point is decreased cytotoxicities. The why polycation is toxic. So one of the reasons is that after internalize into cellular compartment and liberate the DNA, still the polycation migrate inside of the cells and undergoes an unspecific interaction with intracellular components and results in the impaired homeostasis. And if cells are in a impaired homeostasis condition, there should be a very big disturbance in the expression of stably expressing genes, which are called housekeeping genes. Indeed, the cell treated with linear polyethylene imine, there is significant downregulation occurs for the expression of housekeeping genes. On the other hand, the cell treated with PASP-DET, there is almost negligible change in the expression of housekeeping genes. So from these results, we can confirm that PASP-DET scored as a very high biocompatible carrier systems. And we utilize this for the treatment of the lung diseases. So first, we confirm the expression of reporter genes, rispirase, and compared to linear polyethylene imine, our system shows a very significant increased transfection. But this is an in vitro study. Then we moved into in vivo, the treatment of mice by intratracheal administration, and very consistent with in vitro results. We do observe a very significant increase in gene transfection. So based on this model study, we moved into treatment of model disease. that is the pulmonary arterial hypertension. That is a life-threatening disease characterized by progressive pulmonary arterial hypertension. So one of the ways to treat this disease is expand the arteries. So this can be done by administration of peptide called adrenomedulin. However, the half-life of adrenomedulin in the body is very, very short. So you cannot utilize this. You cannot direct injection of adrenomedulin. So our idea is to construct a plasmid DNA expressing adrenomedulin. So in this way, we can expect a continuous secretion of adrenomedulin in the lung to continuously expand the artery to decrease the pressure of right ventricular pressures. So first we prepare the model animals and we confirm the expression of adrenomedulin gene by RT-PCR in animals as shown here. And this is the results. As you can see here, this is a change in the right ventricular pressure. And only in the case of the animal treated with Micelle with adrenomedulin shows the significant decrease of right ventricular pressures. So from this result, we can say that this kind of the non-viral gene vectors really useful for the treatment of this kind of intractable diseases. So because of the time, not so much time left, so if you can allow us me a few minutes, I would like to show you briefly the mechanism. So as I mentioned, this is in case of the virus, the special peptides play a role for the endosomal escape. But our system is more simple. It just undergoes a protonation, single protonation in pH 7 to double protonation in pH 5. So this double protonated structure seems to show the very strong membrane disruption abilities. So how can we confirm this? By the leakage of cytoplasmic enzyme, LDH. So at pH 7.4, in the case of the phosphatase in a single protonated state, there's almost no enzyme leakage. Means very weak interaction with plasma membranes. However, in the pH 5.5, there's significant increase occurs. That means that only at the acidic condition, this polycation shows a strong interaction with endosomal membranes. And this also confirmed by hemolysis assay. At pH 7.4, even increased amine concentration, there is no hemolysis. But at pH 5.5, there is a significant hemolysis with increasing the concentration of amino groups. So that means that this system is quite smart. So at the pH 7.4, almost no toxicity, but only in acidic condition, it shows a strong membrane disruption abilities. And additionally, we recently found out this PASP-DET shows a spontaneous degradation in physiological condition. This is a gel filtration chromatography at 37 degrees, and this is the initial. And after 30 days, it totally degraded. And this is only for the PASP-DET, and if you change this to polyglutamate, there's no such kind of degradations. And we also follow the degradation process by mass spectrometry. And after 3 weeks, you see some monomer signal. And after 2 months, totally degraded into monomer units. And because of this complete depolymerization, we can significantly decrease the cytotoxicities. This is the cytotoxicity against HUBEC. And Xgen 500, polyethylene is quite toxic. And PASP-DET is less toxic than polyethylene, but still toxic. But degraded compound of PASP-DET has almost no toxicities. And after IP injection, there's no induction of inflammatory cytokines. So this is another reason why PASP-DET is very biocompatible. And we can confirm the structure again by TEM. This is a very well regulated rod-like structures with an average size of 58 nanometers. So we are now starting to produce this in the GMP level by a chemical company in Japan and use this in a human clinical trial. So finally, I would like to express my sincere thanks to all of my colleagues, co-workers in the medical school and the pharmaceutical school, and of course, all of my students and colleagues in my laboratories. So thank you very much for your kind attention. Thank you very much.